go ahead and start this. So, urinary system. For the urinary system, you're going to erase this stuff right here. So for anybody that logged in after I gave you these points back, you'll see a +1 off to the side if you missed those. Okay, so for the urinary system, we're going to be looking at filtering blood, and we're going to filter blood so that, and remember the spleen also filters blood, but for very different reasons. Kidney is going to filter blood to remove things like organic waste products. At the kidney, we're going to filter blood so that we can help to regulate how much sodium should be in the blood, how much potassium. So it's going to help to homeostatically regulate solute concentration of that blood plasma, what's in that blood. We're going to also, at the kidneys, as we filter the blood, we're going to work on homeostatically regulating what the blood volume is. By regulating blood volume, we're also helping to regulate how concentrated the blood plasma is, so what the osmolarity is. And by regulating blood volume, we can also help to regulate blood pressure. So we're going to be walking through some hormones that target the kidneys. So we'll look at things like aldosterone and ADH in some more detail. And not today, but on Wednesday, we're going to talk about something that's really important, that absolutely will be on your test. I'll talk to you more about later. And it's going to be one of those things that if you're going into nursing, will definitely keep popping up, okay? And it's called the renin-angiotensin system. We'll get into it later. It's like one of those things you've got to know, right? It's one of those things. So we're going to filter blood. We're going to remove organic waste and other compounds, solutes from the blood. We're going to then form urine in the process, and then we're going to eliminate that urine and all the organic waste and excess solutes that are in there. And while we're doing that, we're going to be homeostatically regulating various parameters. We look at structures. In terms of gross anatomy, very straightforward. Okay, gross anatomy, we've got two kidneys. If you look at the kidneys, they are like the lungs in that nothing comes in or out of these organs, except on the medial side, you've got structures coming in and out of the kidney, just like on the medial side of the lungs, you have structures coming in and out. And that general area where structures come in and out is called the hilus or the hilum. What comes into the kidneys would be the renal arteries. What comes out of the kidneys would be the renal veins, and then these tubes called the ureters. So urine is so we're gonna filter the blood in the kidneys. We're going to form urine in the kidneys, and then that urine is gonna exit the kidneys through the ureters. Those ureters then drain into the bladder or the urinary bladder. And the urine is gonna be stored in the bladder until we're ready to eliminate it from the body. And the way that it's going to exit the body from the bladder is through this tube right here called the urethra. Now, males have a longer urethra, females have a shorter urethra. For males, the urethra runs through the penis. So we'll pick your urethra up again for males when we talk about reproduction. Okay? Because females have a shorter urethra and males have a longer urethra, and because of just the positioning of it, females are much more prone to bladder infections and kidney infections than males are. For males, it just has to travel a lot further in order to cause a problem in the bladder or the kidneys. Where we're going to spend most of our time is microscopically. We're going to be looking microscopically in the kidneys. Before we get to that though, let's do a little bit more gross anatomy as we do a cross section of the kidney. So this right here is a coronal cut, so like this, or a frontal cut. We've taken the anterior part off and now we're looking inside of it. So what we've got is, instead of having a serous membrane around the kidneys, like we saw around the heart, we saw around the lungs, you'll see when you do your GI worksheet, it's around your GI structures. We don't have that here. Here, it's more like what we saw last quarter with the epimysium around muscles, periosteum around bones, the epineurium around nerves, and that is, we just have a fibrous capsule. Dense connective tissue, lots of collagen fibers in it, goes around that capsule. It's not a serous membrane, it doesn't secrete a fluid. Okay? We don't have two layers to it like we've seen all quarter. If we look in the kidney then, the outermost part of the kidney is called the cortex, so kind of like how we saw the adrenal gland, kind of similar to that. And then there's a fibrous capsule on the outside. The outermost part is called the cortex, or the renal cortex. And then everything else, all this stuff here in the middle, is part of the renal medulla. Okay? Now within the renal medulla, there's lots of structures within this middle part, within the renal medulla. You have these kind of triangular or diamond-shaped structures. Those are called renal pyramids. You'll also see them called medullary pyramids because they're in the medulla. Okay? Medullary pyramids. And then in between these renal pyramids or these medullary pyramids, you've got cortical tissue that kind of dips down in here, and that's called the renal column. So you go renal column, renal pyramid, renal column, renal pyramid, column, pyramid, so on and so forth. At the base of those pyramids, so here, down at the tip of those pyramids, there's a little grooved area at the base of those pyramids. And that's where we're gonna see urine first collect, is at the base of those pyramids here. And where that urine first collects is in a structure called the minor calyx. So minor calyx is a little grooved area at the base of the renal pyramid, and that's where urine first collects, is in the minor calyx, or minor calyces for plural. There would be a piece of tissue that covers this that's been peeled back, so we're looking inside of it. Okay. Now the urine that collects in these minor calyces then will drain into a larger groove called the major calyx. So if we look at that major calyx, we would have this minor calyx, and this minor calyx would drain into that major calyx. So major calyx that these minors would drain into, major calyx that these minors would drain into, and then we'd have one like right here that these minors would drain into. And then the urine that's drained into these major calyces then will drain into a larger one called the renal pelvis right here. And then as we exit the renal pelvis, we then go into this tube called the ureter, and then that urine is leaving the kidneys at that point. So urine first collects in the minor calyx that's at the base of the renal pyramids. From there it drains into the major calyx. From there it drains into the renal pelvis. From the renal pelvis, all the urine then exits the kidney through the ureter. That urine goes down to the bladder. It's stored there until we initiate a urination reflex that we'll talk about on Wednesday. And then the urine, if you decide to act on that reflex, the urine will then exit the bladder through the urethra, and away it goes. Okay, so now what we're going to do is we are going to look microscopically. We're going to be looking at a structure called the nephron. So that's where we're going to spend really the rest of our time, is at this nephron. The nephron is the smallest functional unit of the kidney. Just like, let's see, last quarter, we talked about the osteons being the smallest functional unit of compact bone. We talked about a sarcomere being the smallest functional unit of skeletal muscle, or a cell being the smallest functional unit of an organism. So the nephron is the smallest part that you can break the kidney down into, but still have all the pieces necessary to do things like homeostatically regulate, filter the blood, form urine, so on and so forth. So the nephron's microscopic. We're gonna see that it will start up here in the cortex, and then it will dip down into the renal pyramids, and then it will come back up into the cortex, and then it will dip back down into the renal pyramid, and there will be a tube that comes down through the renal pyramid. And what exits that tube will drain into the minor calyx. And what will drain into the minor calyx will be what? What did we just say? Will be what? Start with a U.
Urine.
Urine, okay? Okay. So let's look at that nephron. All right, when we look at these kidneys, we've got about a little over 1 million. Let me back up. So when we look at these nephrons, the nephron is going to consist partly of these little tubes, right? And we have a little over 1 million nephrons per kidney. And if we take the tubes, which are called tubules, of all these nephrons, of all the nephrons in each kidney, we would have about 85 miles of tubule if we stretch them all end to end. So that's a lot, right? 85 miles of these little tubules that are all kind of coiled up as part of a nephron in these kidneys. Okay. Now, I'm gonna come back to this slide in a minute, but I'm gonna put another slide on so we can look at this nephron. We're gonna draw it out, and then we're gonna come back to this slide and talk about how we get blood into the kidney, how we get blood down to the level of these nephrons, how blood leaves a nephron, and how blood leaves the kidney. That's where we're gonna ultimately go here in the next few minutes, okay? All right, so we'll come back to this, okay? And I'm gonna put this up here because this is our nephron right here. Okay, so let's draw this guy. And let me get my markers out of here. And I tell you, at the end of the quarter, I always ask people what their favorite system was over the 2 quarters. And I get tons of people telling me that the endocrine was their favorite and the urinary system was their favorite. And I think a lot of it is because you don't realize all this stuff is happening. There's a lot of really interesting things happening here at the kidney. All right, let's use this one. Okay, so we're gonna see nephron. Nephron is the smallest functional unit of a kidney. It consists of 2 parts. First part is called the renal corpuscle. The second part is called the renal tubule. Now, the renal corpuscle consists of parts. The renal corpuscle consists of Bowman's capsule. Now, you're going to hear me probably call it Bowman's capsule more than anything else. The trend is to move away from names that are due to the original male that found them, right? The trend is to move to a more descriptive term. So though I'm going to probably say Bowman's capsule because that's what I learned, you're going to probably hear it more often called, I'll try to stay black here. You'll probably hear it called more commonly the glomerular capsule. Glomerular capsule. All right, now what the Bowman's capsule is, or the glomerular capsule, It is a hollow ball of cells. So it's formed from a layer of cells, and inside of it is hollow. Okay, so it's a ball that's made of a single layer of cells, and inside of that ball it's hollow. Okay. Inside of that ball it's hollow, and we're going to put something inside of that hollow ball of cells. And what we're going to put in there is a tuft of capillaries called the glomerulus. So the glomerulus, glomeruli for plural, is a tuft of capillaries. Okay, so first of all, what's a tuft? Okay, all this is when we've been drawing out capillaries, you'll always see me do this, right? You always see me say, this is an arteriole, and it takes us into a capillary bed. That's what you've seen me draw. And then leaving that capillary bed, we go into a venule, right? This would be a capillary bed or capillaries, same thing. A bed of capillaries. And around that would be a bunch of cells, right? Here, we just have a tuft of capillaries, just means it's kind of a knot of capillaries. It's kind of think of a cat playing with a ball of yarn, okay? It'd be like a tuft of capillaries like that. And that's what's inside of here. And that should make sense because remember, what the kidney has to do is filter blood. And only at the level of a capillary can things, water, solutes, come in and out of the blood. So pretty good bet that this is where we're gonna first filter the blood, is at these capillaries. Unfortunately, we've got this hollow ball that can collect anything that comes out of the blood. Anything that gets filtered out can collect in that hollow ball right there. Now, we're gonna walk through all of that in lots of detail, okay? But what comes out of the blood, we will call the filtrate, okay? We'll get into that later. It's not urine yet, okay? It's called filtrate. So everything that gets filtered out of the blood, out of the glomerulus, that collects in Bowman's capsule is called the filtrate, not urine yet. Eventually we'll get urine, but not yet. Now, that filtrate that's gonna come out of the blood and collect in Bowman's capsule will then start traveling through the renal tubule. Now, the renal tubule has several different parts. The first part of it's called PCT. Proximal because it's closest to its corpuscle. Proximal convoluted because it's not nice and straight, it's kind of twisty, it's convoluted. Convoluted. Tubule because it's a little tube. Okay, next, that filtrate that's traveling now through the PCT will now travel through what's called the loop of Henle. Loop of Henle. And again, the trend is to move away from persons' names. You'll also see the loop of Henle called the nephron loop. Okay. Then that filtrate will next travel through the DCT. This is distal. Distal because it's further away from its corpuscle compared to the PCT that's closer to its corpuscle. Distal convoluted, again, because it's not nice and straight, it's all tangled up. Tubule because it's a little tube. And then I'm going to put this under the renal tubule, and I'm going to put an asterisk by it because some books will say this is not technically part of the some books will say that this part B is not technically part of the nephron. I tend to put it here in the nephron because what happens with the DCT, a lot of the same things are going to happen at this next part. This next part is called the collecting duct. The collecting duct. And the reason why it's technically, by some books, not part of the nephron is because you will have multiple DCTs from different nephrons draining into a collecting duct. that's why it's often not called part of the nephron, because you don't have one per nephron. You have one per multiple nephrons. Multiple DCTs from different nephrons will drain into a collecting duct. Now, when the filtrate leaves the collecting duct, that's when it's urine, okay? So when the filtrate leaves the collecting duct, that's when we have urine. And where did we say that urine first collects? We said it first collects in the minor calyx. So if we come back here, we're gonna draw this out in just a second. But I'm gonna point to it on here first before we draw it. If we look at this nephron, this part right here is our renal corpuscle. We've got a hollow ball of cells right here that's been cut coronally so we can look inside of it. So this right here is Bowman's capsule. It's a hollow ball of cells. It's formed from a single layer of cells, and inside of it's hollow. Inside of it, we have this tuft of capillaries right here called the glomerulus. And it's gonna be in here that we're gonna filter the blood that's in these glomeruli. We're gonna force water and solutes to come out. And when we looked at a regular old capillary and looked at filtering at the arterial end, the water and solutes came out into the interstitial fluid. But not here, because the water and solutes that come out of this don't go into the interstitial fluid. They're collected in this hollow ball called Bowman's capsule. Okay? Again, everything that comes out of those glomeruli, all the water and the solutes that come out of the blood right here, out of the glomeruli, collects in Bowman's capsule, and what comes out is called the filtrate. Okay? Now that filtrate then will start traveling through the renal tubule. It'll travel through the PCT, then the loop of Henle, which has a descending limb and then an ascending limb, and then into the DCT, and then we're gonna have multiple DCTs from different collecting ducts that come over like this. Sorry, multiple DCTs from different nephrons draining into a collecting duct. As that filtrate travels through this collecting duct, once it exits the collecting duct, okay, so let's say there, once that filtrate exits the collecting duct, it is then urine, and it will collect in the minor calyx. Now, while we still have filtrate, okay, while we still have filtrate, the reason it's not urine yet is because we can still manipulate that filtrate. We can either add more things to the filtrate, like add more solutes to it, or we can take things that are in that filtrate, let's say sodium for example, and put it back into the blood. And we're gonna talk about what those processes are called. But while that filtrate is traveling through the renal tubule, through the PCT, the loop of Henle, the DCT, and the collecting duct, we can still manipulate it. We can still add to it. We can take things from it. We can add to it by taking things that are still in the blood and putting into that filtrate. We can take things that are in the filtrate and put it back into the bloodstream. And we'll look and see where do you do that, because we can't do it here because we've left the glomerulus. So we'll see where we do that in a minute. But as soon as that filtrate leaves the collecting duct, we cannot manipulate it anymore, and that's why at that point it is urine. Once it's urine, we can't do anything with it. We can't add to it, we can't take anything from that urine. All that urine will do is travel from the minor calyx to the major calyx, from the major calyx to the renal pelvis, from the renal pelvis to the ureter, go to the bladder to be stored, and then exit the bladder through the urethra. We can't do anything with it anymore. Okay, we're done manipulating at that point. Okay, now, if we think about where this nephron is, okay? So I said we're gonna start up in the cortex, and then we're gonna dip down into the renal pyramid twice. And I'll show you on another image in a minute, but the corpuscle is in the cortex, the PCT and the DCT are in the cortex. So cortex, loop of Henle dips down into the renal pyramid. DCT, we're back in the cortex, and then the collecting duct dips down into the renal pyramid, so that the end of it is down there at the base of the renal pyramid, and what comes out collects in the minor calyx. So let's go back so we can see how that looks. Actually, I'm going forward 2 slides, actually. Yeah, I'm going forward 2 slides. So here's our kidney. We've got our fibrous capsule. This part up here is the cortex. And then we have renal pyramids. So if we take this right here and put it here, this part right here would be the cortex. And then this part right here would be the renal pyramid. Or sorry, this would be the medulla in here. And within that medulla, within this middle part in here, that part right here is the base of your renal pyramid. So here's the very end of your renal pyramid right there. And we said at the base of that renal pyramid, you have a little groove. That's this right here. That little groove is your minor calyx. So here we're looking at 2 nephrons. Let's just look at this one for now. Okay. So here we've got Bowman's capsule. Here they haven't done a cross section, so we can't look inside of it. But what would be inside of that is that tuft of capillaries. Okay, so inside of there is where we filter the blood. Inside of there is where we have our filtrate, everything that came out of the blood. That filtrate then will travel through the PCT into the descending limb of the loop of Henle, into the ascending limb of the loop of Henle, then go into the DCT, and the DCT drains into the collecting duct, and once we leave the collecting duct, then we have urine, and that urine collects in the minor calyx. Okay. So we have the corpuscle is in the cortex, the PCT is in the cortex, the loop of Henle dips down into the renal pyramids. It's either going to loop way down into the renal pyramids or just a little bit into the renal pyramids, depending on what type of nephron we have. The DCT is back up in the cortex, and then the collecting duct dips down through the renal pyramids and terminates here at the base of the renal pyramids. So that what comes out of the collecting duct is urine, and it collects in the minor calyx. Okay. Now, I'm going to draw this so that we then can talk about how blood comes in and out, okay? And then once we draw blood coming in and out, then we'll talk about what kinds of things can we do to manipulate this filtrate. I keep saying you can add things to it, you can take things out. We're gonna learn what those things are called and how you do that, okay? We're gonna keep it relatively general today, and then later today and on Wednesday, we'll get into lots of detail about how stuff works. All right, so I'm going to erase this, okay? I'm gonna erase all this so I have this whole board to work off of. And I'm going to go back several slides and put this one up, which is looking at how we can get blood into the kidney, how we get blood down to the glomerulus, how we get blood out of the glomerulus, and how we get blood out of the kidney. Okay, that's what we're going to look at next. Okay, so before we do that, we're going to draw the nephron. We're going to draw this. Draw Bowman's capsule. And I will come back and put the glomerulus in when we start drawing blood, because the glomerulus is they're capillaries, so it's part of your blood vessels. So we'll come back and do that in a minute. Okay, so this one is Bowman's capsule. As we leave Bowman's capsule, we go into the first part of the renal tubule, which is called the PCT. Now, the PCT is convoluted, okay? I'm not going to draw it convoluted, so we can make it as clean as possible. So just keep in mind, this can be all twisted here. So that's our PCT. I'm just going to draw a little section of it that's straight. And then from the PCT, we go into the descending limb of the loop of Henle, then the ascending limb of the loop of Henle. So descending limb of the loop of Henle, ascending limb of the loop of Henle, and then we go into the DCT. Okay, so let's label this. Okay, I'm going to say loop of Henle. And I'm not going to write on here right now, but descending limb, ascending limb. And on Wednesday, we'll talk about how those two limbs are different from one another. Okay, it's pretty important, but we'll do that on Wednesday. Okay? For right now, this whole U thing is the loop of Henle, okay? Or also called the nephron loop. Then we go into the DCT, and again, remember it's convoluted. I'm just going to draw a little section that's straight. And then from the DCT, we would then drain into the collecting duct. And remember, we would have multiple DCTs draining into a collecting duct. So I'll just do it like that. Now we'll put a CD here for collecting duct. All right, so now we're going to draw blood. Okay, we're going to start thinking about how we get blood coming in and out of this thing. Okay, so first, we're going to think about how we get blood coming in and out of the kidney, first of all. Okay. So, let's put it up here. Okay, so leaving the heart, leaving the left ventricle, blood would next go into the what? Come on. The aorta, okay. Don't make me start to have heart palpitations first. Okay, so we're gonna leave the left ventricle, we then go into the aorta. Remember, the aorta has different parts to it, right? We've got the ascending aorta. So we'll come off of the top of the heart. And then we would go into the aortic arch. And then, actually, let's just draw it correctly. So we would go from the left ventricle, we would go into the ascending aorta, and we would go into the aortic arch, and then we would go into the descending aorta. Descending aorta has different parts to it. First part of the descending aorta is the descending thoracic aorta. That's just the part that runs through your thoracic region. And then once it crosses your respiratory diaphragm, then it becomes your descending abdominal aorta. Now, coming off of the descending abdominal aorta, you've got your left and right renal arteries. And that's how you get blood into your right and left kidneys. You've got a right renal artery that's going to go into your right kidney. You have a left renal artery that's going to go into your left kidney. So that's how we get blood into each kidney. Right there. All right, so here we are. Here's our renal artery. Okay, and if we look over here, here's our renal artery. Now, within each kidney, that renal artery is going to branch into more numerous but smaller diameter arteries that we're not going to learn the names of. And then those arteries within each kidney are going to branch into smaller diameter but more numerous arteries that we're not going to learn the names of. And then those arteries within each kidney are going to branch into smaller diameter but more numerous arteries that we're not learning the names of. And then those arteries within each kidney are going to branch into smaller diameter but more numerous arteries that we're not learning the names of. And And then those arteries are gonna branch within each kidney into smaller diameter but more numerous, very small arteries called arterioles that we are gonna learn the names of. So, coming into the glomerulus, we come in through an arteriole. So ultimately, this renal artery would have branched into smaller diameter, but more numerous arteries until eventually we get into these arterioles. So 1, 2, 3, 4. 1, 2, 3, 4. 4 that we're not learning the names of, okay? But eventually, we get down into a little tiny artery called an arteriole. And arterioles take you into capillaries, okay? That's what we've learned so far. And the capillary that this arteriole takes us into is a capillary called the glomerulus. So remember, the glomerulus is a tuft, kind of a tangled mess. And again, to keep it kind of neat, I'm not going to draw it as a tangled mess. I'm just going to draw it as a backward C. This in here. Okay, so we've got an arteriole taking us into the glomerulus. Glomerulus is inside a Bowman's capsule, and the glomerulus is just a tuft of capillaries. Now, so far, what you guys have learned is arterioles take you into capillaries, and then what would take us out of a capillary? What's that kind of vessel called that takes blood away from a capillary? A venule. But that's not what this is, okay? That's not what that is. Normally, that's what we would have, but that's not what that is. That thing that's taking blood out of the glomerulus, this thing right here, is another arteriole. So that's a little weird, okay? We haven't seen that before. It's a little odd. So, and we'll see why that's the case, but that's what we have there. And since we've got two arterioles, one bringing blood in and one taking blood out, we have to name them so that we don't confuse them, because they do very different things. One brings blood in, one takes blood out. So the one that's bringing blood into the glomerulus, we're going to call that one the afferent arteriole. Okay. And the one that takes blood away from the glomerulus, we're going to call that one the efferent arteriole. And now, based on what you guys have learned, what would be the guess of where blood's going to go next? We're in an efferent arteriole, so where would you think blood would probably go next? Into what kind of vessel?
The artery?
From an arteriole, you typically go into a an arteriole usually takes you into a capillary, right? An arteriole usually takes you into a capillary, right? So what we've learned so far is you've got arteries. They branch into smaller diameter but more numerous arteries until eventually you get down to little teeny tiny arteries called arterioles. Those take you into capillaries. Then you leave capillaries through little tiny veins called venules. And then venules drain into larger but less numerous veins. And then eventually you get back to the heart. Okay? That's what we've learned so far. So what we've learned to date is arterioles typically take us into capillaries. Okay? So, here it's a little bit weird. We have the afferent taking us into a capillary, the glomerulus. And now we've got this thing called an efferent arteriole, which we haven't seen anything like this yet. But based on what we know, arterioles should take us into capillaries. And that's actually where we're going to go next. We're going to go into another capillary. So this is, again, weird. We haven't seen an an arterial, a capillary, arterial, and a capillary. We've never seen that, but that's what we got here. Okay? Now the question is, where are those capillaries that the efferent arterial is going to take us into? The capillaries that the efferent arterial take us into are called the peritubular capillaries. Okay? Now the name tells you where they are. Peri means around, tubule, around the tubule. The peritubular capillaries are wrapped all around this renal tubule, okay? All around the renal tubule. Now again, for sake of making it as simple and clean as possible, I'm gonna limit how much I have it wrapping around here, but I'll show you what it kind of looks like. So here are your peritubular capillaries. These guys, they wrap all around the tubule. Those are your peritubular capillaries. So that little thing right there is your afferent arterial taking you into the glomerulus. There's your efferent arterial taking you out of the glomerulus. And that efferent arterial then goes into these peritubular capillaries that are all around your tubule. So again, here's your afferent arterial taking you into the glomerulus. Your efferent arterial takes you out of the glomerulus, and then into the peritubular capillaries that wrap all around the tubule. Now again, that should kind of make sense because what I've been saying is that while that filtrate travels through the tubule, the PCT, the lymphotherapy, the DCT, I keep saying we can manipulate it. I keep saying we can manipulate it. We can either add things to it or take things out of it. And when I said that we could take things out of the filtrate, I said we could put it back into the bloodstream. So anything that comes in or out of the blood can only do so at the level of which one of these blood vessels? Where can oxygen only come out? At a capillary. Where can sodium only come in or out? At a capillary. Where can water only come in and out? At a capillary. Only at capillaries can things come in and out. So again, it kind of makes sense to wrap capillaries around here because if we're going to take things that are in the filtrate and put it back in the blood, we need to have a capillary right there where it can enter the bloodstream. Okay. All right, so let's draw these peritubular capillaries. Again, I'm going to keep it real simple. And do it like this. Maybe too big. Wrap it like that. One more wrap around here. Okay, so all that's for peritubular capillaries. So write it up here. Next, go into the peritubular capillaries. Okay, so again, all of that wrapping around the tubule are the peritubular capillaries. So then, let's then think about, as we leave the peritubular capillaries, what would make the most sense that we would go into next, based on what we know to be true about the circulatory system? Venules, and that's what we do. Now we're kind of back to normal. Then we go into venules. Okay, now let's come back over here. So here's our afferent arterial taking us into glomerulus. Leaving the glomerulus, we go into an efferent arterial. From an efferent arterial, we go into peritubular capillaries. And now we're leaving the nephron region and we go into venules. Okay? Then those venules are going to drain or converge within the kidneys into veins that we're not going to learn the names of. And then within each kidney, those veins are going to drain or converge into larger diameter, less numerous veins that we're not going to learn the names of. And then within each kidney, those veins are gonna drain or converge into larger diameter but less numerous veins that we're not learning the names of, until eventually all the blood within that one kidney will exit the kidney through the renal vein. So here we go, 1, 2, 3. Okay. Let's go. 1, 2, 3. And then we exit the kidney, exit each kidney through the renal vein. You have a right and a left, okay? You exit the left kidney through the left renal vein. You exit the right kidney through the right renal vein. And these 3 veins between the venules, 2, 3, and the renal vein, we're not learning the names of those. And then the renal veins are gonna drain into what large vein that's gonna be heading back to the heart? Drains into the right atria. What's that large vein called?
The vena cava?
This one right here.
Vena cava. No, the acid.
S comes this way.
Inferior.
The inferior vena cava. Inferior vena cava, okay, is what the renal veins drain into. Renal veins drain into the IVC, and now we're back at the right atria of the heart. All right. So now, I'm gonna ask you guys if you have any questions in a minute. But then we're gonna start thinking about what kind of processes are taking place between the nephron and the blood that's flowing in and out of the nephron. Okay, we're gonna talk about those processes. Before I do that, I just wanna make sure that we're all kind of on the same page thinking about this. If you have a little over 1 million nephrons per kidney, okay? If you have a little over 1 million nephrons per kidney, how many glomeruli do you have per kidney? It's not a trick question. If you have a little over 1 million nephrons per kidney, how many glomeruli do you have per kidney?
A
Little over 1 million, okay? And if you have a little over 1 million nephrons and glomeruli per kidney, how many afferent arterioles do you have per kidney? A little over 1 million. And if you have a little over 1 million nephrons, glomeruli, and afferent arterioles per kidney, about how many efferent arterioles do you have per kidney? A little over 1 million. And about how many peritubular capillaries do you have per kidney? A little over 1 million. And about how many venules do you have per kidney? A little over 1 million. Okay, all right. All right. So this is happening a little over 1 million times in each kidney, okay? What we're drawing here, there are a little over 1 million of those per kidney. Now, if you want to take a picture of that, go ahead, because I'm going to start adding to this. Okay, so let's find the right slide that we should be on right now. Okay, so let's see. We are going to talk about filtration very generally, and we're going to get into a lot more detail with it later. Okay? We're going to talk about reabsorption and secretion. And for right now, what I want to do is just say where these processes occur, between what, and in which direction is water and or solutes moving. That's what I want to think about right now. And then later we'll talk about the mechanism of how it happens, what forces determine this, what kind of transport mechanisms, things like that. Okay? So first of all, filtration. Let's think about filtration. When we looked at a regular old capillary, okay, so let's kind of go back and review. When we looked at a regular old capillary, this is the way your book and I drew it. It was like this. So this part right here was the arterial. And this would be just a regular old systemic capillary. Okay, the arterial would bring blood into the capillary, and this was our capillary here. And then this would be a systemic capillary. And then blood would leave that systemic capillary and go into a venule. Okay? And when we looked at just a regular old systemic capillary, we said that if you looked over the course of an entire day, on the arterial side of that capillary, what you saw was that the blood pressure in that capillary, which was called the capillary hydrostatic pressure, forced water and solutes out. Okay, that's what we said. We said if you looked over the course of an entire day at the arterial end of a systemic capillary, water and solutes were forced out. That was called filtration. Okay, we called that filtration. And the driving force for this filtration was the fact that there was blood pressure in that capillary that we called the capillary hydrostatic pressure. Capillary hydrostatic pressure is the blood pressure in the capillary and it forced water and solutes out. We said if you looked over the course of an entire day on the arterial end, you would have something like 30 liters of fluid coming out of your systemic capillaries at the arterial end and coming out into the interstitial fluid. Things that couldn't come out would be things that were too big, right? Things that were too big to cross the wall of the capillary. Name one thing that should not leave, that should never leave your blood and end up in your interstitial fluids because of capillary hydrostatic pressure. What should be too big to get forced out because of that blood pressure in your capillary?
Albumin, isn't it?
Albumin, a plasma protein, right? Plasma protein should be too big to cross this wall. When you do get albumins ending up out here in the interstitial fluid would be when you have really high blood pressure, that could cause that potentially. But normally your plasma proteins like albumins should be too big to be able to get forced out. Okay, so they should always stay in your blood. Your big old plasma protein should be in here. Okay? What else should be too big to get forced out? What's the biggest thing in your blood? What's bigger than a protein? It's a structure that contains proteins.
Hemoglobin? No.
What's the hemoglobin in?
Red blood cell?
Oh, a cell, right? A cell. Cells are bigger than proteins, right? Please tell me you know that. Yes, I know you know that. I'm just playing with you. So cells are made of proteins, carbs, and lipids, right? And they are, if a protein is too big to cross this wall, then a red blood cell or white blood cells should be too big to be forced out. Now remember, white blood cells can shimmy their way out, but that's not because of this. That's just because they can do kind of cool things. But cells should stay in the bloodstream, and big old plasma protein should stay in the bloodstream. So then, but most of the water and solutes that are coming through this capillary, boom, they come out. Okay, they're forced out because they're small. And because the pressure in here is high enough to force them out because the wall's thin. Okay, that is filtration. But we said that if that water and solutes stayed out, that we would die from this, right? Because one, we would have all of our water from our blood out here, and all of our solutes like glucose and sodium, all that stuff would be out here, and that would cause our blood volume to do what? Drop dramatically, right? And if our blood volume dropped dramatically, what would happen to our blood pressure? It would drop too. You would die from that. Plus, you would have so much edema or swelling, you wouldn't be able to move at all. So fortunately, here at the venial end, most of that comes right back in. And if it doesn't come right back in, it'll end up in capillaries that are close to this, called what kind of capillaries? Lymphatic capillaries. It will become lymph. Okay, good. So, what drives that water back into here? What drives the water back? Blood colloid osmotic pressure. This is saying osmotic pressure draws water back in, and we're not looking at all solutes, we're looking only at those big old plasma proteins. That's the BC part, blood colloid. That's the big old plasma proteins. So osmotic pressure is the driving force of water to move from high water to low water, or from low plasma proteins, where there should be none out here, to high plasma proteins in here. That's BCOP. Okay, so BCOP. And actually, we'll go through this all again on Wednesday when we do that, maybe today even. Okay, so that's what draws the water back in. Okay, so this we're gonna see happen here as well, just with a few minor differences. So we're gonna talk about all these forces in detail over here in a little bit, not right now. What we want to talk about right now is just the fact that filtration happens here, okay? All of that was just to say, in here, like here, blood pressure is going to force water and solids out, okay? And that happens in Bowman's capsule. So in Bowman's capsule is the site of filtration, okay? Filtration happens in Bowman's capsule. Infiltration is water and solutes getting forced out of the blood, in this case, out of the glomerulus, into Bowman's capsule. And this hollow space in here is called the capsular space, right? It's just the empty space in Bowman's capsule. So water and solutes get forced out of the glomerulus, out of the blood, into Bowman's capsule, into the capsular space of Bowman's capsule. And this water and solutes that come out, we're going to call it now the filtrate. That's where we produce filtrate, is right there in the renal corpuscle. The renal corpuscle is the site of filtration, and that's all we do there. We don't do reabsorption, we don't do secretion. We only do filtration. Okay, now, again, the driving force for this is not diffusion, it's not concentration gradients. We're not using pumps, we're not using channels. What drives the water and solutes out is blood pressure in the glomerulus. The blood pressure in the glomerulus forces water and solutes out, and anything that's small enough to cross this wall will, okay? Not through pumps, not through channels, there's little pores in there, okay? We're gonna learn that these glomeruli are leakier than a regular old capillary. Okay, and that helps water and solutes to come out. They've got more leaky holes, basically, pores, than these guys do. We'll talk about that later. They're called fenestrated capillaries. Okay, they're leaky. Yeah.
So just filtration happens there, not the BCOP?
Not, yes, but we will, the BCOP is, yes. All of this is happening here, and the net result of all this is water and solutes come out. Okay. So yeah, we will talk about later, we'll tease this apart and see exactly what's all going on there. So it'll be blood pressure and BCOP, and the net effect will be water and solutes come out, even though some water comes back in because of BCOP. Okay, so we will come back to that. So the overall process, when you talk about filtration, happens only at the corpuscle. Okay, no pumps, no channels, no concentration gradients, no active transport, only blood pressure driving water and solutes out.
Can you just repeat it when you said that H2O and solids get forced out? I didn't get to it the rest time.
I don't know what I said. Anybody want to tell me?
You said it like like 3 different ways, that's why I was like, I don't know what's the best way to
Water
write
and
it.
solutes get forced out because of blood pressure in the glomerulus. That process is called filtration. Does not involve pumps or channels. Does not involve concentration gradients. The driving force for that is pressure.
Thank you.
Yeah, yeah, sorry. Okay, so now we have filtrate, okay, now we have this fluid. Now, before we go into reabsorption and secretion, the other two things that could happen on here, I'm gonna get rid of this because we don't need this anymore. I'm gonna make sure that we are fine with the terminology of these different fluids, okay? So, water and solutes that come out of the glomerulus that collected Bowman's capsule filtrate, okay? As that filtrate starts traveling through the renal tubule, it's also called the tubular fluid. As this filtrate starts traveling through the PCT, the Lupus-Henle, the DCT, the collecting duct, it's also called tubular fluid. It's the fluid in the tube. The filtrate, when it starts traveling through the tubule, is also called the tubular fluid. So we'll say filtrate as when it travels through the tubule, which is the PCT, the Lupus-Henle, the DCT, and the collecting duct. It'll also be called the tubular fluid. I'll call it filtrate, okay, but the slide you'll see tubular fluid, the book might reference tubular fluid as well. Okay. Now let's think about out here. Out here is just what? What's this out here?
Medulla.
Interstitial fluid, that's all this is out here. Okay, so the interstitial fluid, of course they're not gonna call it interstitial fluid, right? That would be way too simple. They're gonna call it the peritubular fluid. Peritubular because it's around the tubule, okay? So out here, okay, the extracellular fluid or the interstitial fluid is called the peritubular fluid because it's around the tubule. Okay. Now, if I pointed in here, if I pointed to the fluid that's in the peritubular capillaries, what would you tell me is in here? It's not a trick. Filtrate is in here, right? What's inside of a capillary?
Blood.
Blood, okay? That's what's in here, blood, okay? What's in your blood vessels is blood, right? What's in the glomerulus?
Blood.
What's in the peritubular capillaries? Blood. What's in your venule? Blood. What's that blood consist of? Water, solutes, cells, proteins, okay? So, if I point out here, you're gonna tell me that this is called the peritubular fluid. If I point in here, you're gonna tell me in there is
Blood.
blood. And if I point in here, you're gonna tell me that's called the
Water. Filtrate.
filtrate or the fluid in the tube is called the tubular fluid, okay? All right, I'm not gonna leave this until we're all on board with that, okay? If you're not, tell me, take a drink of water. Where's filtrate formed? And it comes out of the glomerulus. It's formed in the corpuscle, right? In the renal corpuscle is where we form the filtrate. The filtrate comes from water and solutes being forced out of the glomerulus, out of the blood. Okay, out of the blood. Okay. All right, now, what we're going to see is, kind of like when we looked at a regular old capillary, way too much water and solutes were forced out, right? We looked down here, we said way too much water and solutes are filtered out of the blood, and we had to get some to come back in, okay? What we're gonna see is, over the course of a whole day, actually, I can't remember the numbers. Yeah, I kind of can. If you look over the course of an entire day, it's something like, if you look at all of your, as that filtrate starts traveling through this tubule, before it leaves the collecting duct and becomes urine, okay? So I'm gonna put a line here. Before it leaves this collecting duct and collects in the minor calyx, and we go from being filtrate to urine. Before that happens, we have to put about 99% of that 48 gallons that we form has to go back in the blood, okay? So, while this filtrate is traveling through the renal tubule, we have to put most of it right back in the bloodstream. We have to put it back in the bloodstream. That's what we're going to talk about first. That's called reabsorption. So reabsorption. Later we'll worry about exactly what it gets reabsorbed where, but we just want to talk in a general sense right now, and we'll fill in details later about what this thing is called reabsorption. Okay, so we're going to do secretion and we're going to do reabsorption. And both of these processes, I'll do reabsorption first and secretion, but both of these processes are going to take place between the filtrate that is in the tubule, or the tubular fluid, and the blood that's in the peritubular capillaries. Okay, I'm gonna say that 2 more times because you've got to get that in your head. Reabsorption and secretion are gonna take place between the filtrate that's in the tubule and the blood that's in the peritubular capillaries. Reabsorption and secretion are going to take place between the filtrate that's in the tubule and the blood that's in the peritubular capillaries. Whereas filtration only occurs where? At the renal corpuscle. With what capillary? The glomerulus, okay? You will never do filtration with the peritubular capillaries, but we will do reabsorption and secretion with the blood that's in the peritubular capillaries and the filtrate that's in the renal tubule, okay? So out here, we're gonna do reabsorption and secretion. Here, we do filtration, okay? Filtration is always forcing anything that's small enough, water and small solutes, out of the glomerulus, out of the blood, across the wall of the glomerulus, into the capsular space of Bowman's capsule. That's how we produce filtrate. Now that we have this filtrate, we need to put most of it back in the bloodstream. We're not going to put it back in the glomerulus. We're going to put it back into the blood at the peritubular capillaries while the filtrate is traveling through the tubule. Okay, so reabsorption is going to look like this. Let's say that we're looking at the filtrate here at the PCT. Okay, so I'm just going to put F for filtrate. Okay, let's say we're looking at the filtrate at the PCT, and let's say we're looking at glucose. Okay, so I'm gonna put some glucose in here. I'm gonna put some sodium in here. I'm gonna put some water in here. This is just filtrate, okay? And it's got a bunch of solutes in it, right? It's water and solutes. Now, let's say that we want to specifically put glucose back in the blood, okay? Because we definitely don't want to leave this glucose in the filtrate, because ultimately it will leave the collecting duct and all that glucose will be in the urine, and now it's gone. We don't get to use it. We wouldn't want to do that, right? So all this good stuff, we want to put it back in the bloodstream, okay? So we do that through a process called reabsorption. So glucose is going to get transported either with pumps or channels. Okay, glucose tends to use a pump, not a channel, but it's going to get transported across the wall of the renal tubule. We'll talk about the details of that later. But glucose will get transported across the wall of the renal tubule. Here we happen to be looking at the PCT. It will then come out here into the peritubular fluid, and then from the peritubular fluid, it will diffuse into the blood that's in the peritubular capillaries. That process of taking solutes, or water, from the filtrate somewhere in the renal tubule and putting back into the blood that's in the peritubular capillaries, that is reabsorption. That process is reabsorption. Okay, I'll say more about that in a minute. Now, kind of the point of all this is to filter out bad stuff, right? To filter out organic waste. Filter out things that we don't need in the blood. But the problem is, the way that filtration works here, we are going to filter out a lot of good things too. We're going to filter out glucose that we're going to have to put back into the bloodstream through reabsorption. We're going to filter out here vitamins that we're going to have to reabsorb back into the bloodstream. We're going to filter out way too much sodium that we've got to reabsorb back into the bloodstream, and so on and so forth. So that's why we're doing reabsorption, because way too much good stuff gets filtered out right here, okay? So reabsorption allows us to put all that good stuff back into the blood so that it doesn't end up in the filtrate and then end up in the urine, or the filtrate that leaves the collecting duct and becomes urine. Now, what is going to happen is not all the bad stuff will have gotten filtered out at the renal corpuscle. So there will still be some things that are in the blood, in the peritubular capillaries, that we actually don't want in there, that we need to get out. Thank you. If anybody can bring this back down, that would be fabulous. Thank you so much. So there will be some things, particularly we'll talk about things like hydrogen ions that affect pH. Some things will still be in the blood that we don't want to be in the blood, that we want in the urine, that didn't get filtered out here. And that's why we do the process called secretion, which we'll go over next. So secretion, I'm going to do it over here at the DCT, which is not to say that reabsorption only happens at the PCT and secretion only happens at the DCT. That's not true, and we'll talk about exactly where these things occur later, okay? But if we wanted to secrete something, let's say hydrogen ions. Let's say that we had a lot of hydrogen ions in the bloodstream, and it was making our blood pH too low, and we didn't want that many hydrogen ions in there. Then we could secrete or do secretion. Secretion is going to go in the exact opposite direction as reabsorption, but between the same things, between the filtrate that's somewhere in the tubule and the blood that's in the peritubular capillaries. It just goes the opposite direction. So here we would have those hydrogen ions let's say they diffuse out, and now they're out here in the peritubular fluid. So they diffuse out of the blood in the peritubular capillaries. They're now out here in the peritubular fluid, and then using, since it's hydrogen ions, we'd have to use pumps or channels. With pumps or channels, we would then have it go across the wall of the tubule and into the filtrate that's in the renal tubule. So this direction is secretion. So reabsorption's taking from the filtrate and putting it back into the blood that's in the peritubular capillaries. Reabsorption's taking from the filtrate and putting back into the blood that's in the peritubular capillaries. Secretion is taking from the blood that's in the peritubular capillaries and putting it into the filtrate so that we can eliminate it in the urine. Okay. These are the three processes that we're going to talk about. So filtration only happens at the corpuscle. Reabsorption and secretion can happen throughout the filtrate that's in the renal tubule and the blood that's in the peritubular capillaries. Filtration only happens from the blood that's in the glomerulus, because it only happens at the corpuscle. Reabsorption and secretion happen between the filtrate that's in the tubule, the blood that's in the peritubular capillaries. Now, adding a couple more details onto this, okay? Questions before I do that? Yep.
Why do you have to take the hydrogen ions from the
So
capillary?
maybe the pH is too low. So the kidney and the lungs are the two places you can homeostatically regulate pH, okay? We'll talk more about that later. But it could be, maybe you're on antibiotics. Maybe you're taking penicillin. And maybe the metabolite or the penicillin, I don't know how that breaks down, didn't get filtered out here. You gotta get that out of your blood. And so then maybe you secrete it. Could be any number of things that ended up still being in the blood that came out.
Okay, thank you.
Okay, so I said over here, for filtration, the water and solutes don't use pumps, they don't use channels. And that's because whatever is small enough will get forced across the wall of the glomerulus, because the glomerulus is very leaky. It's got little pores in there. So we don't use pumps and channels there. It's just whatever's small enough will get forced out. When you look at the cells that make up the renal tubule, they are mostly simple cuboidal epithelia. Some places it's simple columnar epithelia, but definitely too thick for diffusion to happen across, okay? So things that are, actually I shouldn't say that, that's not necessarily true. But over here, we're not gonna have leaks and pores in the renal tubule, I'll say it that way. The cells are simple cuboidal epithelia or simple columnar epithelia that line this whole tubule. And if we want to get solutes or ions, things that are water soluble, to go across the wall of the renal tubule, we have to go across the simple cuboidal epithelia or simple columnar epithelia, which means you have to cross at least two plasma membranes, okay? So let's say, for example, let's say that we're looking here. Let's say we're looking right here. Let's say right here we've got simple cuboidal epithelia. Hydrogen is an ion, right? So that means it is not lipid soluble, it is water soluble. So if we want to take these hydrogen ions and get them into the filtrate that's here in the collecting duct or VCT, we have to cross this plasma membrane right here, and then that plasma membrane right there. So that means we have to have either a channel or a pump for hydrogen here, a channel or a pump for hydrogen right there. So when we do secretion and reabsorption, if what is moving, if what is being secreted or reabsorbed is water soluble. So for reabsorption and secretion, for water soluble molecules and or ions, we have to use pumps and or channels. Right? Because things that are water soluble can't freely cross the plasma membrane. If we use pumps, then we can either be doing active transport, or we could be doing carrier-mediated diffusion. Just depends on what it is and where it is. We'll get into this kind of detail later, okay, once we start talking about specific things that get secreted or reabsorbed. If we're dealing with channels, then the only thing that ever happens through a channel is diffusion. You can't do active transport with a channel. So channels, you're always going to be doing channel-mediated diffusion. Which means now when we start thinking about reabsorption and secretion, now we're worrying about concentration gradients. Where are things high? Where are things low? Unless we're talking about active transport, then we can move up a concentration gradient if we apply ATP or some kind of energy source to it. Anything that's moving through reabsorption or secretion that's lipid soluble, so things like fat soluble vitamins, urea, I can't remember if that's a lipid or not. I can't remember. But things that are lipid soluble, these things can only diffuse across the plasma membrane. You can't actively transport these things that are lipid soluble. They only diffuse, and they don't use pumps or channels. They don't need to. Okay? Things that are lipid soluble just freely cross the plasma membrane. That's called simple diffusion. Simple diffusion, when you diffuse, but you don't use pumps, you don't use channels. So that would be things like oxygen, carbon dioxide, fatty acids, fat soluble vitamins, ethanol. Ethanol would be a good example here, ethanol. And then water, right? So water will only move passively. You never actively transport water, ever, okay? Water only ever moves passively via osmosis. So osmosis says that water will move from areas where you have high water to areas where you have low water. Or what we're gonna see a lot here is we're gonna see that water is gonna follow solutes. So if solutes are moving in some direction, then water will follow it, because water will go from low solutes towards high solutes. Right? So if we start reabsorbing glucose, water's gonna follow that. Water's going to passively follow that. Now, with water can either freely diffuse across the membrane, so it either can do simple diffusion, which again is just basically osmosis. Actually, let's say it this way. We can do osmosis either with a channel or not. We'll say it that way. You can do osmosis with a water channel or not. Water doesn't need a channel, but if there are channels for water, it will diffuse faster. Okay? water channels are called aquaporins. Okay, I'm reviewing this. This should have been stuff that you did in bio and A&P 1, okay? But just in case we have a gap in there, I wanna make sure that you remember that stuff. And if you've forgotten it, it's not a bad idea to just go do a review of transport. So we're gonna be talking about transport for this week and next week. Okay, so. When you go to study, one of the first things that you wanna do is make sure when I say the word secretion, you automatically have an idea what direction the movement's occurring. So secretion is gonna be taking from the blood, putting into the filtrate. Second, you wanna automatically be thinking where it's happening. So it's gonna be happening somewhere between the renal tubule, the filtrate that's in the renal tubule, and the blood that's in the peritubular capillaries. If I say reabsorption, again, you wanna immediately think about the direction. Reabsorption is putting back into the blood, and you wanna think about where it's happening. It's gonna be somewhere between the filtrate that's in the tubule and the blood that's in the peritubular capillaries. And we'll flush out details along the way, okay? But those words, you need to automatically know the direction, and in the grand scheme of things, where. Versus filtration is always moving in this direction, only here at the corpuscle. Filtration never is worrying about concentration gradients, pumps or channels. Reabsorption and secretion are, because most things that we're gonna be reabsorbing or secreting will be water soluble. Some aren't, but most will be. Okay? So, if there were no glucose pumps here at the PCT, you would not be able to reabsorb glucose at the PCT. Okay? If there were no glucose pumps at the PCT, you would not be able to reabsorb glucose at the PCT. So we're gonna have to talk about what kinds of things get reabsorbed and where. Does glucose get reabsorbed at the PCT? Happens to, yes, which then automatically tells you there are glucose pumps in those plasma membranes. Whereas we're gonna learn that glucose does not get reabsorbed at the loop of Henle. That automatically tells you there are no glucose pumps in the cells at the loop of Henle. So that stuff will start filling in. Okay, questions on that? Okay, all right. So let's do a few more things here. So we covered this. We've actually covered multiple slides. I just didn't kind of click on them as we were talking. Okay, so let's say a couple things about the fact that there are different types of nephrons. Okay, so we're gonna look at two different types of nephrons and talk about how are they different from one another and why do we have two? Okay. So you've got what are called cortical nephrons and juxtamedullary nephrons. Now I want you to be careful with this word juxtamedullary, because later we're going to talk about a structure called the juxtaglomerular complex, which is very different from juxtamedullary nephrons. Okay, I don't want you to get the words confused. Cortical nephrons versus juxtamedullary nephrons. For humans, we have about 85% corticals and about 15% juxtamedullaries. Other organisms will have different ratios depending on what they need, and we'll take a guess at what those other ratios might be in just a minute once we know where we're at. First of all, cortical nephron. The main difference between a cortical and a juxtamedullary is cortical nephrons, most of the nephron is in the cortex. Okay? A cortical nephron, most of it's in the cortex, which means when you look at the loop of Henle, its loop of Henle is real short and it only dips down into the medulla just a little bit. It's a short loop of Henle. Versus juxtamedullary nephrons have really long loops of Henle. They dip way down into the renal medulla. That's the big difference right there. So if we look at them, this is what we looked at before. cortical nephron has a real short loop of Henle, well, actually, the part of the loop of Henle that dips down into the renal medulla, or the renal pyramid, is just a little bit. Most of the nephrons up here in the cortex. Versus if you look at these juxtamedullaries, you can see they sit a little bit lower, but they have a long loop of Henle that dips way down into the renal pyramids. Okay, that's the big difference between the two. Now, what is the functional significance of this? Structurally, that's how they're different. Corticals have a short loop of Henle dipping down into the renal pyramid a little bit. Juxtamedullaries have a longer loop of Henle that dips way down in the renal pyramid. What we're going to see is there are lots of places as the filtrate is traveling through the renal tubule, PCT, loop of Henle, PCT, collecting depth. There are lots of places and different mechanisms where we can reabsorb water. We want to make sure that we reabsorb a significant amount of water so that we don't end up losing too much water to our urine. If we didn't reabsorb enough water and we lost too much water to the urine, that would make our urine output really high and our blood volume would become very low. If we did not reabsorb enough water, our blood volume would get too low because our urine output was so high. And that could start to influence our blood pressure. Okay, so that's the first thing. It would also make us too dehydrated. We would not want to lose too much water because we would start to get dehydrated. So there's multiple mechanisms that we're going to learn about that make sure that we reabsorb a fair amount of water. One of those, one of many, is the fact that we have these nephrons and their loops of Henle dip down into the renal pyramids. And we're going to see that at these loops of Henle, whether they're shorter in the corticals or longer in the juxtamedullaries, one of the key things that we do here at the loop of Henle is called countercurrent multiplication. We'll talk about that on Wednesday. But what we're going to see happens in countercurrent multiplication is we are going to reabsorb sodium, chloride, and then water will passively follow. And it's a beautiful mechanism that we'll learn the details about on Wednesday. But that's what we're going to see as one of the major things that happens at the loop of Henle. We're going to take sodium and chloride that's in the filtrate, put it into the blood that's in the peritubular capillaries, and then the water follows it. Now, if we look at the corticals versus the juxtamedullaries, for humans, having 15% juxtamedullary gives us the right amount we need with these other mechanisms that we have to make sure that we reabsorb enough water. When we look at those loop of Henles, the fact that the juxtamedullaries have a longer loop means that there's more surface area in the juxtamedullary loops of Henle. They're longer, more surface area, more places to reabsorb water. The other important thing that we're going to talk more about later is when you look out here in this peritubular fluid, what we're going to see is up here where the cortex is, the osmolarity out here is about 300 milliosmoles. And when you come way deep in the renal pyramids, it gets greater. It's about 1200 milliosmoles down here. Now, first of all, if we're comparing 300 versus 1200, which one has more solutes in it? 300 or 1200? 1200, okay? There's more stuff in here compared to water. This is less stuff. Okay. Now, why do we care about that? We care about that because water doesn't just magically come out of the filtrate and into the peritubular fluid. Water only ever moves via osmosis. Water only ever moves towards a high concentration of solutes. So if I asked you, where are we going to have more water coming out of the filtrate? Here where it's 300 or here where it's 1200? Which one?
1200.
Or it's 1200. The greater that concentration gradient, the more that the water will move, the faster diffusion will happen. So if we've got a really long loop of Henle versus a really short loop of Henle that only comes here, the long loop of Henle is gonna allow us to reabsorb more water. We're gonna pull more water out. And once we get the water out here, where's it gonna go next? It's gonna go into the blood that's in the peritubular capillaries. It will get reabsorbed. Okay? So those long loop of Henles, they are really important in making sure that we produce a nice concentrated urine, making sure that we don't lose too much water, making sure that we have reabsorbed a lot of water. If we're reabsorbing a lot of water, then our urine becomes nice and concentrated so that we're not losing water to the urine. If we had none of those juxtamedullary nephrons, we would lose too much water. Our urine would not be concentrated enough. We would not reabsorb enough water. We would lose too much water, and we would end up being dehydrated all the time. Now, if I were to tell you, comparing a human to a camel, are camels gonna have more or less juxtamedullary nephrons compared to a human?
Yes.
Do camels have to deal with environments that are more likely to cause them to be dehydrated or less likely to cause them to be dehydrated compared to humans? More. So they are ultimately concerned about losing too much water. They really need to make sure that they've got lots of mechanisms in place to reabsorb a lot of water so they don't lose too much. So they're gonna have more or less of these long loop of Henles. Do we want them to reabsorb more water or less?
More water.
More. Which ones reabsorb more, longs or shorts?
Lungs.
Long, so they would have more or less juxtamedullaries compared to humans.
Less.
They would have more, because they need to reabsorb more water. The juxtamedullaries are the ones that allow them to reabsorb more water. Think about it later, okay? They would have more. All right. Okay, let's take a 5-minute break just to shake everything off. And then what we're gonna do, I'm gonna go until 8:30. I know that's painful, but I'm gonna do that. What we're gonna do is we're basically gonna start at the corpuscle. We're gonna walk our way through the nephron and just talk in a relatively general sense about what kinds of things are happening where. So we're gonna talk about reabsorption happens here versus there. What kinds of things get reabsorbed here versus there? We'll talk about epithelial cells here versus there. We'll talk about those kinds of things. And then on Wednesday, we're gonna come back to the corpuscle. We're gonna go through the whole nephron all over again, but do it in detail, okay? So let's take a 5-minute break and then talk for another 30 minutes, and then we'll go upstairs. I'm gonna keep the, also running, so things are being recorded, so keep that in mind. We'll go over the details of it later, but basically what's gonna happen here, if we look at the loop of Henle, okay, what we're gonna see is that water is going to, well, let's see, how do we wanna say this? Let's just look at water, okay? If you look at the osmolarity of out here compared to in here, there's more stuff out here than in here, and we'll talk about how does all that work later, okay? But for now, we'll just say there's more stuff out here. And the deeper that you go in the medulla, the more stuff is out in the peritubular fluid, okay? Closer that you are to the cortex, the less stuff's out here. Still a lot of stuff, but less. So in both of these cases, whether you're looking up here or you're looking down here, water's gonna come out because of osmosis. So water's gonna do that. and water's gonna do this. But between the two, you're gonna get a lot more water coming out down here than you will here, just because of osmotic pressure. The driving force is greater because you have a greater concentration difference down here than you do up here, okay? And then once that water's out here, then it will, pretty quickly, it will diffuse into the blood that's in the peritubular capillaries. Now remember, there would be lots of them. They'd be all over the place, right? So the water will eventually end up going into the blood that's in the peritubular capillaries. And when we look at coming from the filtrate, going into the peritubular fluid, into the blood, that is reabsorption. This is reabsorption. It's just that down here, we reabsorb more water than we do up here. Does that make sense?
Yes.
And by reabsorbing more water, we're ending up with more water in the blood and less water in the filtrate. And by the time that we get over here, we're gonna actually see this happen other places as well. But by the time we get over here, what we're gonna have here is that filtrate is gonna have little water, and we will have put a lot of water back into the blood over the course of all this, so that this urine here is gonna be very concentrated. It's gonna have a high osmolarity, it's gonna have a lot of stuff in it and little water, relatively speaking. And that makes sure that we don't get too dehydrated. It makes sure that we don't lose too much water, that we put enough water back into the blood. How will you get like a concentrated water?
Yeah, it's a separate question.
Okay, yeah, go ahead.
Okay, so for reabsorption,
Uh-huh.
does it have like the pumps or no?
It depends on what's getting reabsorbed. So it could be a channel, it could be pumps, it could be active transport, it could be just carrier mediated diffusion. It kind of depends on where you are and what's getting reabsorbed. And we'll fill in some of those gaps on Wednesday, okay? We'll talk a little bit about it today, but we'll fill in more on Wednesday. So it just depends. So like water never uses a pump ever. It only will move through water channels, or it'll just freely come out. But if there's water channels, it can come out easier. Okay? It can diffuse faster if there's water channels. Versus something like glucose is just a bit too big for a channel, so it always uses a pump. And if you look over here, because you have so much glucose here, because we just filtered all that glucose out. So now you've got tons of glucose in the filtrate and very little in the blood. So now you have a big concentration difference. And you have, so you've got a lot of glucose here, very little in the blood, and you have glucose pumps. You're gonna have carrier mediated diffusion for that glucose here, okay? Versus if you look over here, you really aren't reabsorbing glucose over here because you did basically all of it here. Okay, you just don't have a big concentration difference over here anymore. And you don't have glucose pumps over here either. So it kind of depends on where the pump is, where your concentration differences are. It'll depend on those things.
Okay, thank you.
Okay, is that good? Okay, all right, good. All right, let's go ahead and walk through this stuff. So this slide here, there's some things on this slide that you guys don't need to worry about. So let me emphasize what I do want you to know from this slide. So renal corpuscle, site of filtration, we've already gone through that. When you look at the renal corpuscle, it consists of Bowman's capsule, or the glomerular capsule, and inside of it is a tuft of capillaries called the glomerulus, you know that. What I want you to know about this stuff is that Bowman's capsule is, for the most part, simple squamous epithelia. Okay, there's a little bit more to it than that, but what I want you to have a sense of is that the cells here are really small, single layer of cells. And that's gonna help to facilitate, let me back up. When you look at the glomerulus, let me back up and say it again. When you look at Bowman's capsule, it's gonna be composed of simple squamous epithelium. Okay, we'll leave that there. And then, when you look at the glomerulus is a capillary. As all capillaries, they're not much more than simple squamous epithelium. Okay? These particular capillaries, the glomerular capillaries, are fenestrated capillaries, which just means that they are leakier than a regular capillary. They're still simple squamous epithelia, but they have a bunch of little pores in them. And that's gonna make filtration happen even more effectively. There's little slits or little pores where anything that's small enough will cross. Okay, that's what I want you to know. You don't need to worry about these podocytes, these pedicels. It's just looking more at kind of how the epithelia is kind of organized here. And if you're interested, you'll see an image on the next slide, but I want you to not get kind of bogged down in all those words and focus more on the fact that these glomeruli are leakier than a regular capillary. They have pores or filtration slits so that anything that's small enough that can easily get through those pores will, okay? The things that should not leave the glomerulus, the things that should not be able to get through those filtration slits or those pores are things that are big. So plasma proteins should not be leaving the blood. You should not have plasma proteins in your urine. And this is why they check your urine for protein. You shouldn't have albumin in there. Sometimes you do. If you have albumin in your urine, you shouldn't. That could mean a variety of things. It could mean that your blood pressure is too high and it's forcing proteins across the wall of the glomerulus where they shouldn't be. It could just be, and that could be a problem or not. It kind of depends on how severe it is and how long that goes on. It could just mean that you have an infection, a little infection, and the capillaries are getting leakier than normal. That's just part of your immune response. And that could be why you have proteins in your urine. So proteins in your urine could mean that you have kidney damage, or it could mean you got a little infection. It kind of runs the gamut. You also should have blood in your urine, right? Because red blood cells should not be able to get across the glomerulus. They should be too big to fit through those pores or those slits. So again, if you have blood in your urine, that could be the sign of something really major or something minor. You've got a little infection, you've mounted an immune response, things are a little bit leakier than normal. Okay? But you shouldn't have blood in your urine because red blood cells shouldn't be getting across the wall of the glomerulus. That's where it would happen right there. Okay, so that's what I want you to know from this slide. I want you to know that your capillaries are fenestrated capillaries. They're very leaky. Anything that's small enough will get through those leaky capillaries. They have pores or slits in them. Things that are small will cross. This right here is just looking at those podocytes, those pedicels. If you're interested, you can take a look at this. Again, I just don't want us to get bogged down on the details of that and lose the big picture. So this right here is telling you that the driving force for filtration, what is ultimately causing filtration to occur, is blood pressure. Okay? The blood pressure in the glomerulus is forcing water and solutes out. The higher your blood pressure, the more water and solutes that will come out, and potentially the more of other things that shouldn't be out, like plasma proteins, like cells. Larger solutes should not be able to leave. Plasma proteins should not be leaving. Cells shouldn't be leaving. What should be leaving are things that are small, like metabolic waste. Excessive ions will enter the capsular space, the hollow space of Bowman's capsule, and become the filtrate. But like we said, all these metabolic waste leave, but so do all these good things. Glucose is going to get filtered out. Free fatty acids are going to get filtered out. Amino acids, vitamins, things we don't want to end up in the urine. Things that we need to put back into the blood. And that's why we're going to do reabsorption, to put this good stuff back into the blood. So as we leave the corpuscle, we then start in on the renal tubule. Renal tubule starts at the PCT. If we look at the PCT, it is composed of simple cuboidal epithelia with microvilli on the apical surface. And that microvilli on the apical surface is there to increase surface area. And that's because we have to do a lot of reabsorption. We said that of all this filtrate we produce, 99% of it has to get reabsorbed before that filtrate leaves the collecting duct in his urine and we can't touch it anymore. And where most of, not all of by any means, but where most of that reabsorption happens is right there at the PCT. That's where we do the majority of this reabsorption. So if 99% has to get reabsorbed, sometime during the renal tubule, sometime as it's traveling through the renal tubule, about 70% of it gets reabsorbed at the PCT. Now, that's not the only thing that happens here. You can secrete here as well. But on the test, if I say, what's the primary job of the PCT, you're gonna tell me reabsorption. It's not the only job, but it is the primary job. And to help facilitate that reabsorption, we increase surface area with microvilli. More microvilli, more places to cross. Okay? Now, what kinds of things are gonna get reabsorbed at the PCT? Organic nutrients. So those would be things like amino acids, glucose. This is where we're going to absorb them, which again tells you that at the PCT, we're gonna have pumps for glucose here. We're gonna have pumps for amino acids there, so we can do carrier-mediated diffusion. We've got lots of amino acids in there, we've got lots of glucose in the filtrate. Pumps, because they're a little bit too big to fit through a channel, we then can do carrier-mediated diffusion and get those organic nutrients reabsorbed back into the bloodstream. Some ions will get reabsorbed as well. We're not going to worry about which ones, but some ions get reabsorbed. If any plasma proteins did get filtered out at the corpuscle, we can reabsorb some of them back into the blood at the PCT, and then water will passively follow all of these organic nutrients and ions that are getting reabsorbed. Water will then follow those. So as glucose and amino acids and ions are getting reabsorbed, water will then follow. Because again, water moves towards high solutes. So as we're reabsorbing solutes, water just passively follows it. Now, next week, we're gonna look at a slide of the kidney, and you're gonna have to be able to tell the PCT from the DCT. It's actually pretty easy to do, but one thing you're gonna look for is the microvilli on the apical surface of the PCT, which you don't have on the DCT, okay? That'll be one thing you look for. The PCT's overall diameter is larger than the DCT. That'll be another thing that you look for. Okay, the loop of Henle. The loop of Henle, we have a descending limb that goes down towards the medulla, and the ascending limb comes up towards the cortex. And when we look at each of those limbs, they will have what's called a thick and a thin segment. Now, we'll look at that more on Wednesday when we talk about the process that happens there called countercurrent multiplication. What we're gonna see is that the thick versus the thin segment will have different types of epithelia, and what's permeable at one area will not be permeable at the other. So some things can cross in the descending limb, other things can get reabsorbed or cross at the ascending limb, and it's because of different permeabilities and different types of epithelia. We will do that on Wednesday, okay, when we talk about the whole process. What we're gonna see at the loop of Henle, or the nephron loop, is we're gonna do this thing, like I said, called countercurrent multiplication, and we're gonna look at sodium, chloride and water passively follow and getting reabsorbed. This is what we're gonna see happen in the loop of Henle. So sodium, chloride, and then water will passively follow, okay? So sodium, chloride, and water, we're gonna see get reabsorbed here at the loop of Henle, and we'll see how that whole process works through this thing called countercurrent multiplication. We'll do that on Wednesday. Now, that's not the only thing that happens there, but that's what we're gonna talk about. That's what we're gonna focus on. Now, when you look at the PCT and the loop of Henle, you're gonna hear me on Wednesday keep saying that these two areas, you do bulk reabsorption. You're gonna hear me say that over and over again. What I mean by that is, we have a lot of reabsorption to do. Of all this filtrate, we have to reabsorb 99% of it. And keep in mind that this filtrate is moving fast through the tubule. And if we don't reabsorb this 99% of it, by the time it leaves the collecting duct, then we don't get to it. It's gonna be lost to the urine and it's gone. So what we're gonna see happen is, at the PCT and the loop of Henle, we're gonna see these two areas kind of be like factories. They're just gonna be pumping this stuff out. Not literally pumping, it'll diffuse, okay? But we're gonna be basically just reabsorbing as much as we can. Bulk reabsorption, okay? We're not gonna worry about fine tuning anything. We're not gonna worry about any other mechanisms coming in to regulate what happens here. We're just gonna have bulk reabsorption based on concentration gradients. We're gonna have a lot of stuff in the filtrate that wants to get back into the blood, and it's just gonna be diffusing, lots of it, okay? Going from the filtrate into the peritubular fluid, into the blood, okay? By the time that we get to the DCT and the collecting duct, now we need to start fine tuning what we need. Because remember, at the end of the day, we need not the filtrate, not the urine, but the blood to be in homeostatic range. We need to make sure that what's in the blood has the right amount of sodium in there. We need homeostatic levels of calcium in the blood. We need homeostatic levels of potassium in the blood. We need homeostatic blood volume. We need homeostatic blood pressure. So at some point, we need to see what's going on in the blood and exactly how do we need to fine tune reabsorption and secretion to make sure that what's in the blood is what we need homeostatically. And that's gonna take place at the DCT and the collecting duct. When we learned about hormones that influence what happens in the kidneys, hormones are gonna either work at the DCT or the collecting duct. This is where we fine tune. We fine tune with hormones. Hormones are gonna tell the DCT and the collecting duct, not exclusively, but a lot, of how much to reabsorb, how much more sodium needs reabsorbed down here to get the blood exactly where we need it to be. So the PCT and loop of Henle, we just reabsorb a lot, okay? We have a lot to reabsorb, just get as much done as possible. So that by the time we get over here to the DCT and the collecting duct, now we can say, okay, now let's see how much more sodium needs to be reabsorbed. How much more calcium, a lot or little? And that will be dependent on things like aldosterone levels, calcitonin levels, PTH levels, ADH levels, okay, things like that. And we're gonna look and see how those hormones affect the DCT and the collecting duct, okay? But I always, you'll hear me say bulk reabsorption, PCT, loop of Henle, fine tuning with hormones, DCT collecting duct, okay? All right, so at the DCT, diameter smaller than the PCT. The DCT, like the PCT, is simple cuboidal epithelia. I don't know if it says there, but it is. The epithelia lacks microvilli for the most part here at the DCT. Again, because even though we are reabsorbing, we're not doing that bulk reabsorbing where we need all that extra surface area that we needed in the PCT. We just don't need it in the DCT. Now, at the DCT, there are gonna be some kind of special structures that are associated with the DCT. One of those special structures is called the juxtaglomerular complex, or you'll also see it called the juxtaglomerular apparatus. Earlier I said, don't confuse juxtamedullary nephrons with juxtaglomerular complex. They're two different things. The juxtaglomerular complex is a group of cells that are right by the DCT, and in this group of cells, there are some endocrine cells in this complex that produce EPO. We talked about EPO earlier in the quarter when we talked about blood. There are a group of cells in this juxtaglomerular complex that produce TPO. We talked about that at the beginning of the quarter. If you don't remember what EPO is, it's on your comprehensive study guide, so if you've forgotten it, you'll learn it again, or you'll learn it for the first time. Regardless, you'll learn it. But what we're going to talk about later is there are some cells within this juxtaglomerular complex that are going to produce not a hormone, but an enzyme called renin. And this is that thing I was talking about that I said absolutely will be on your final. It's one of those things that if you're going into healthcare, particularly nursing, you're going to see this thing pop up again and again. On your nursing board test, it's going to pop up. It just pops up in pathophys. It's going to pop up again and again, and that's the renin-angiotensin system, okay? Renin is an enzyme. It's going to work on something called angiotensin. We will walk through all that later, okay? It's one of those step by step by step by step by step things. We'll walk through it. It will absolutely be many points on your final exam, okay? I don't know, at least 5, if not 8, okay? It'll be a chunk. We'll get to that later. Okay, now what happens at the DCT? 3 important things, not the 3 only things, but 3 important things. 1, we will do active secretion of certain ions. We'll talk about which ones later. Acids, this is your hydrogen ions, affecting your pH. Certain drugs, so like if you're taking penicillin or some antibiotic, and various toxins, okay? Now, I want you to think about what this word means. These 2 words, active secretion. Secretion tells you in what direction things are moving. So secretion tells you that these things are in the blood, like acids, like hydrogen ions, and we need to get it out of the blood and into the filtrate so that it ends up in the urine, okay? Active tells you how we're gonna do it. Active tells you what kind of transporter we're gonna use. Active transport, okay? So we're gonna use active transport pumps to pump various ions, acids, drugs, and toxins out of the blood and into the filtrate. We're gonna use active transport pumps, okay? We'll talk more about that later. Also at the DCT, we will do selective reabsorption of sodium, calcium from the tubular fluid, from the filtrate. So again, I want you to think about what the words mean. So first of all, reabsorption tells us that we're gonna take certain, let's say sodium. We're gonna take sodium that's in the tubular fluid, in the filtrate, and we are gonna reabsorb it, put it back into the blood that's in the peritubular capillaries. That's reabsorption. We're gonna do that with sodium, and we'll do that with calcium. Selective tells us we're only gonna do this if a hormone tells us to do it. That's the selective part. So what hormone would tell the kidneys to reabsorb sodium? Which one? Aldosterone, okay? So depending on what our aldosterone levels are, will dictate how much sodium gets reabsorbed at the DCT, okay? What hormone would tell now you know the DCT of the kidneys to reabsorb calcium so that we're losing less of it? What hormone is gonna try to get calcium up in the bloodstream, reabsorbing it, putting it back in the blood? Which one?
Oxytocin.
Not ADH, PTH, PTH. PTH is going to cause calcium to get reabsorbed here at the DCT. Then down here, the last major thing that's gonna happen here, again, not the only thing, but the last major thing, selective reabsorption of water. Okay, so let's go back to aldosterone for a second. Aldosterone is gonna cause sodium to get reabsorbed. Depending on what aldosterone levels are, will dictate how much sodium gets reabsorbed, and then water will passively follow it. So with aldosterone, you reabsorb sodium and water passively follows it. Okay. But there's also another hormone that works here at the DCT and the collecting duct that only causes just water to get reabsorbed. It doesn't do anything with minerals. What's that hormone? That's ADH. Okay, that's ADH. So depending on what our ADH levels are will dictate how much just water gets reabsorbed here. We'll be reabsorbing water as we reabsorb sodium, that's aldosterone. But also we'll be reabsorbing just water under the influence of ADH. Okay? And we'll talk more about that later. Okay. Now, lastly, we're gonna wrap up here in just a couple minutes. The collecting duct, or the collecting system. Typically, if you look at the epithelia, it's simple columnar here. And again, you're gonna see me put the DCT and the collecting duct kind of together. Because some of the things that happen at the DCT can also happen at the collecting duct, and vice versa. So I tend to think of these two areas together, but technically they are separate. Okay, but I tend to think of them together. But the collecting duct is the last place that we have to do reabsorption or secretion. Once that filtrate leaves the collecting duct, it is then urine and we can't do anything with it. Okay, so that's our last place to make any alterations. And again, remember, what we ultimately are trying to do is not get the urine in homeostatic range. We're trying to get the blood in homeostatic range. And whatever needs to end up in the urine, so be it, so that we get the blood where we need it to be. So that we get the right amount of sodium in the blood, the right amount of calcium in the blood, the right amount of water in the blood, and so on and so forth. Okay. All right. Then, okay, last slide. This right here is just looking at urine production and metabolic waste. So again, the goal is to maintain homeostasis of the blood, okay, of blood volume, of blood pressure, of blood sodium levels, of blood potassium levels, blood calcium levels. When we do this, what we will end up seeing is by the time we're done doing all of this reabsorption and secretion, getting the blood where we need it to be, we're gonna see that the urine is very concentrated. Okay? When we start this whole process, the filtrate basically looks like the plasma minus there are no plasma proteins in it. Otherwise, what comes out pretty much looks like the plasma. So the concentration of the filtrate and the concentration of the original blood plasma is pretty much equal. But as that filtrate travels through the tubule and we secrete and reabsorb, by the time that filtrate leaves the collecting duct, it does not look like blood plasma anymore. It should be way more concentrated than blood plasma, so that again, we're not losing too much water. We want that water back in the blood so that our urine's nice and concentrated. Now, you might be thinking, I always hear that you don't want your urine too concentrated, right? It's all relative. When I talk about urine being 4 times more concentrated in your blood plasma, if you were to look at the color of this urine, it should be pale, like so pale yellow that it's barely yellow. If your urine looks yellow or orange, It is way too concentrated. If your urine is this color or darker, you need to drink more water, okay? And mine's always this concentrated. But 4 times more concentrated should be very concentrated, but still be clear almost. When your urine starts getting a lot of color to it, it means that you're having to reabsorb so much water that the urine becomes way concentrated. And over time, you know, that could put you at risk for, you know, kidney stones and all kinds of things that you don't want, okay? You don't want to do that to your kidneys too much. You don't want to have that super high concentrated solutes running through there. Remember, there's organic waste and nasty stuff in there. So 4 times concentrated compared to your blood plasma is where it should be. If it's too dark of a color, it's way, way too concentrated. 3 major metabolic wastes I want you guys to know. Urea, this will be your most abundant one. This you get mostly from your liver taking amino acids and turning them into other things. When you break down amino acids, and the liver is very good at breaking down amino acids and turning them into other things, you have to take off, well, we won't get into the metabolism of it, but urea is a waste product that you form. We'll just leave it at that. Okay? Urea is a waste product that you form from your liver taking amino acids and turning them into other things. Creatinine. We talked about your skeletal muscles using creatine phosphate to generate molecules of ATP really quickly. If you're doing a lot of really intense strenuous activity, you will be producing a lot of creatinine, and that's a waste product. And then uric acid's the other very common one. Uric acid you get from recycling nitrogenous bases of DNA and RNA, uric acid. Okay, so this right here is just a summary of what we basically been talking about. On Wednesday, we will start back at the corpuscle. We will spend a lot of time here talking about all the different forces, BCOP, hydrostatic pressure, that determines whether we produce a filtrate or not. Because there will be times where we don't produce a filtrate. And for short periods of time, that's okay. But if you don't produce a filtrate, you ultimately won't produce what? If you don't produce filtrate, you ultimately will not produce a what? You won't produce urine, okay? And for short periods of time, it's okay if you don't produce filtrate. But if you're not producing filtrate for extended periods of time, and you're not producing urine, that's bad. Okay, so we're gonna have to talk about how do we regulate this. Okay, we're gonna spend a lot of time here. And then we'll move through as much as we can on Wednesday, talking about details. There'll be a couple things we'll have to wrap up the following week, just a few things. Okay, I'm gonna shut things down. I'm gonna go upstairs.